Both blastocyst biopsy and vitrification have been a revolution in IVF, in vitro fertilization, allowing embryologists from all around the world to perform preimplantation genetic testing on human embryos with minimal risk for the embryo.
Trophectoderm biopsy has allowed the retrieval of about 7 cells from a fully grown embryo, thereby enabling robust downstream genetic analysis. Moreover, no impact has been shown to date for this approach.
The clinical efficiency of this workflow allows patients with monogenic conditions and all those patients with increased risk of chromosomal abnormalities within their blastocyst to benefit from preimplantation genetic testing.
There is still room for improving the current strategies of embryo selection. For instance, myenomic and proteomic profiling represent intriguing options to complement preimplantation genetic testing on a single trophectoderm biopsy.
Inexperienced operators often struggle with opening and penetrating the zona pellucida. It's just a matter of focus. The blastocyst, laser objective, and pipettes should be on the same focal plane.
After labeling the biopsy dish with the patient's details with a permanent, non-toxic marker, number each 10 microliter drop of Heaps buffered medium with the embryo and cycle ID. Using a 300 micrometer stripping pipette, transfer a viable, fully expanded blastocyst to the first drop of the biopsy dish and rinse the drop to remove the excess culture medium before moving the blastocyst into the second drop in the dish.